technologies were, what are the current technologies, what are the things you have to think about when you're doing library construction, because a lot of you are analyzing the data. I can do that. And then show you a couple things that sort of, the analysis of sequencing data is important based on how you do sequencing. All right, so what sequencer is the best? This has been not as big of a problem anymore since the number of sequencers on the market have reduced significantly. What are the things you need to think about when you're doing library construction? And some of the things about sequencing quality that impact what you do. So the different sequencers. So this was that sequencer I was just talking a little bit about, is the helicose sequencer. And this is just to tell you how did the helicose people actually had a patent on this sequencing by synthesis chemistry. And so how this worked was, and the power of the helicose was that in this particular type of sequencing, you were able to actually capture mRNA directly on the sequencing flow cell. And so this flow cell had a bunch of T's on it. It's actually an oligo-GT capture flow cell. And what happens is the poly A tail gets captured, and then we initiate cDNA synthesis and put a poly G tail on this with terminal transferase. And then this allowed you to then use a poly C primer to initiate sequence from the 5' side of the mRNA. This is a pretty powerful technique because you could directly analyze cDNA. They eventually developed direct RNA sequencing with this. But the main feature of all these sequencing platforms was this thing, which is you can visualize, in the helicose sequencer, you can visualize each cDNA molecule on the flow cell. So it was literally single molecule sequencing. So you would add your G nucleotides, take a picture, and each one of these is a cDNA being sequenced. And that's what these dots are, and that's how all the sequencers work. Take lots of pictures as you add an A, T, C, or a G. So that's why the data files, which you guys probably will never see, are enormous, because there's just picture after picture after picture, and you need to know location. Now, in the helicose and the Illumina and the ABI sequencer, the main issue is if you have dots that are too close to each other. And then those are what we call, like in the Illumina platform, it's your cluster density is too high. And the reason why is you can't distinguish this strand from this strand in the Illumina sequencer. So the main distinguishing thing for helicose was this single molecule sequencing thing. No amplification, no bias for amplification. You sequence what is native. And so that's why it cost $1 million, but it died. So the ABI, imaging-wise, works very similar, but the ABI, which is the sequencer that we got from ABI, which was then bought by LifeTech and Thermo Fisher, and this sequencer died too. So we had 3 of these taking up, basically taking up a lot of space in the sequencing room, but the platform is completely dead because Illumina killed it. Or ABI, Thermo Fisher killed it. This one worked by imaging again, but this time instead of adding individual base edits at one time, you would add doublets. So you would add a double A, a double AC, and so this is what we call color space. So you had this 4x4 color space coding system, and what they would do is you add a doublet, and then you would add another doublet on top. And what that allowed you to do was then you would get 2 base calls on every read. Does that make sense? Every nucleotide you get double call. And so that was the thing that they sold. They sold that we are more accurate than Illumina because we get double calls on the same base. Does that make sense?
Was it faster as well?
It was not that much faster. Sequencing by ligation, it's actually a little slower. But they sort of sold higher accuracy. And their reads were a little longer, but at the end of the day, Illumina crushed them because of market share. So another issue was this is like C-FASTA data. So the data is like in this kind of space. So analysis platforms, we're dealing with FASTA files or FASTQ files, which you guys are playing with now. This is a totally different type of data output. And so I think that's ultimately what killed this platform, is computational people, most of them were dealing with FASTA, FASTQ files, and no one was making computational bioinformatics or bioconductor packages for CFASTA, even though there are some significant advantages for this type of chemistry. Now, this type of chemistry is still being used, but not in this ABI 5500 XL. I don't have a slide on this. I don't know if you guys have seen this in situ seq, George Church's lab. So they can take a slide with tissue, or cells really, and they can sequence the cell, the nucleotides in the cell as you're imaging it. So you can actually sequence the transcripts in situ. It's pretty awesome. But they use this kind of chemistry to execute that. So this chemistry platform is still viable. The guy that did that is now, he's now a faculty at Cold Spring Harbor. And so this is the type of chemistry that is still used for those kinds of sort of next level technologies that are, you know, cool but difficult.
So on a quick note on CS-FASTA, if you're ever looking at that data, you want to align it using bowtie 1. Top hat 2, bowtie 2, none of the aligners work. Star does not work. The only aligner that does alignment for CS-FASTA is bowtie 1, the previous version of bowtie. Okay.
Yeah.
Right.
So, and you know, that's one of the limitations of that, but again, from a technological standpoint, it has a lot of advantages over this, which is Illumina. So Illumina, HiSeq, MiSeq. So Illumina has 3 sequencers, HiSeq, MiSeq, and NextSeq. So HiSeq, MiSeq are the same chemistries, okay? And the way that it works is you have what we call a universal primer here called P5 and another one called P7 on the other side of all of your nucleic acids. The P5 binds to the sequencing flow cell, and P7 does, and it creates these what we call bridges, and this is then amplified, and then as you make these, they make what we call clusters, okay? So you just keep doing this amplification, now you have a cluster. So this is different than helicose because helicose took one molecule and imaged it, right? This is, you make a big dot of the same clone, and then you sequence it. But the idea is the same. So the camera is taking a picture of each of these clusters instead of a single molecule. And so the advantage for the Illumina was, well, you know, we had the ability to have a camera that didn't need to image a single molecule anymore. You could image a larger thing. And so that's what made this sequencer faster. I made it a little more accurate from a sequencing call standpoint because, you know, you had a larger dot.
Is it larger or brighter?
It's larger and brighter because it's a physical clusters. It's like many.
But is it a fraction limited spot or no? I'm just.
I don't know how.
It's still smaller than the resolution of light, I'm assuming.
Yeah, it's still small, but it's not. It's much bigger than a single molecule, obviously. So this is like maybe I don't know how many molecules are in there each cluster, but no. But that's what made. There's all these lawsuits between Helicose and all the sequencing companies because Helicose claimed that sequencing by synthesis with cameras was their IP. They ultimately lost in court, but you know, you could sort of see like all these sequencers ran the same way, except for some of the basic chemistry stuff.
So, you know, David, this whole idea of how the clustering happens is not very well. It's not. They don't talk about it very clearly.
They don't talk about it very well.
And I ran into several problems.
Yeah.
When I was using small RNA sequencing.
Right.
What we ended up discovering was that if you are using an older version of the adapters, they're not compatible with the new machine.
Right.
And when you look at the adapter sequences and where they claim complementarity is required in order for you to get proper clustering, you realize that the old ones should work, but in practice they actually don't. What they don't tell you, for example, is that the sequence that's right next to the P5 and P7 is also very important because clustering does not just involve 2 regions that are complementary finding one another. It actually involves a synthesis step, which they don't even talk about, which is dependent on the sequence that's right next to those.
Right.
So you have to be very careful about how you're designing it and whether it's compatible with your machine. And because you don't have access to a lot of the background information, and most of the people that work at Illumina don't even know this. You really have to go talk to the top people, which is what I ended up having to do.
Right, you have to talk to the upper level technical people.
Absolutely, people that have been with the company.
So that was the other difference between ABI, Illumina, and Helicose. So Helicose, we knew exactly what the chemistry was. so we can modify it for our purposes for circulating tumor cells. ABI, same thing. That's why ABI, the ABI chemistry, people were able to use for in situ seq. Illumina, Illumina is now talking to the in situ seq people to like modify for their sequencer, but that's the problem is that this is like a little bit of a mystery, but we do know what the P5 and P7 sequences are. People have constructed their own custom libraries, and it does work. I've done that. It does work, but there's some voodoo that we don't understand. But anyways, the bottom line is that high cluster density, you get overlapping clusters. That's the problem when you load too much, and so that's why you always have to include. You always have to quantify the amount of input you're putting in. The second thing that is hurtful for sequencers all like this is if you have the same sequence, the sequencer cannot deal with the same sequence over and over again. So it needs complexity of what they call nucleic acids for sequencing. Because if this is all red, all at the same time, the sequencer has a very bad problem with that. So, so sometimes they ask you to have high complexity DNA. So for RNA-seq, it's usually not a problem. For DNA-seq, it's usually not. But if you're doing like amplicons, like 20 amplicons, this thing will crash on you. So they usually have you add in what we call Phi-X, which is just some sort of artificial synthetic nucleic acids that bind and add complexity for sequencing imaging. And these are all these like little.
Is that just for focusing?
It's for focusing, exactly. And this is for base calling too. Because if you just see all red, if you see the same sequence, sometimes it's harder to resolve individual clusters.
So the reason why this whole chemistry and process has never made sense to me is because they don't actually, it's like intentional, or is it supposed to make sense?
No, I think it's proprietary, and I don't know, like, you know, unlike ABI and Helicos, which I, you know, talk directly with the scientists, and they just tell me what's on the, you know, we were able to modify those protocols for our purposes. Yeah, it's a black box. Yeah. All we know is they show these little pretty clustering things, and there's P5, there's P7.
And what is the P5 and the P7?
It's just a sequence. I'll show you. It's just a sequence. It's a known sequence that they made. And so there's different ways to run this sequencer. Like other sequencers, you can do single end reads. Usually about 50 to 150 base pairs is good. You go to 300, they have kits for that, but you know you start seeing sequence quality issues of over 100 base pairs. For the high-seq, you have 7 open lanes per flow cell. Most cores use 6 of those. They use one of them as a loading control. So and there's 2 flow cells per run. So that's about 200 million reads per lane. So you have usually about you have about 6 lanes of sequencing, and 6 lanes times 200 million is 1.2 billion reads. Okay, so it's close to a human genome. So it's usually a high-seq run if you do a couple of genomes. And so the core is about $1,900 per lane. This takes 6 days to run, so it's like launching the space shuttle. They do have a rapid run mode, and so the cores have this option where they can take 2 lanes, so 120 million reads. You lose the number of reads, but you can run this in 24 hours, and they charge a little more for it. So most of investigators are interested in this sort of situation. They pay a little more, but you only have to fill 2 lanes. Here, they have to wait till they fill 12 lanes. Right? That's a whole lot of 1.2 billion reads is a lot of reads for a single investigator, unless you're a genome person or a probe. Okay, and so here they have 4 different colors: A, T, C, and G. Okay, and so just for you guys to know, it's just like everyone else. There's these 4 colors. Now NextSeq was different. Okay, so it looks same clustering, but the difference here is, for all of you to know, is that they use T as green, C as red. A is the hybrid, and G is no signal. Okay? So the G no signal thing gets people, right? Because it's like, wait, you say you're calling a base pair based on no signal. Okay, but that makes it very fast because now you only have 2 colors of chemistry to an image, right? So it reduces the sequencing time theoretically in half. Much faster, but slightly higher error. Okay, so the mutation call people sometimes don't like NextSeq because of that sequence chemistry difference. Does that make sense? You get 400 million reads here for about $2,500 with that's just the reagents. We have our own NextSeq here at the Cancer Center, which all Cancer Center investigators have access to. It's not a core facility, so someone would have to load it for you. For most of the Cancer Center PIs that are me, Haber, or Wetstein, it's Toshi Shioda that does that. But but the cost is, so remember it's 200 million reads maximum on the HiSeq for about 2,000. You're getting about 400 million reads for you're basically getting half the price for the reads when you do the NextSeq. Okay, and that's a combination of it's not core and sequencing chemistry is cheaper. And this runs overnight. So you can get this kind of data in 24 hours. They have this mid-output thing for sort of people that don't want to pay as much for more reads, but most people don't do this. I mean, what did they tell me? The people that would want to do this are the yeast people. So,
No, I use the high output.
Right.
I never use the high output.
So that's the population they're talking about, small genome people would focus on this. Well, you're Mo, so you can do whatever you want. So, alright, so the other sequence, so I told you about two sequencers that are dead. Illumina, MySeq, HiSeq, NextSeq are alive. PacBio is still alive. So PacBio is a very different sequencer. We at Cancer Center technically have access through Howard Hughes, through Daniel Haber. There's a facility at UMass that runs the PacBio there for Howard Hughes investigators in Massachusetts. The largest number of PacBios are probably in Manhattan. Mount Sinai has probably the most number of PacBio representatives and sequencers. But this is like a very different sequencer. So in some ways it's very similar to Helicose because it is single molecule sequencing again. There's no amplification bias because of that. Extremely long reads. So it's basically has a polymerase here and it's imaging as it's synthesizing. And that's how this sequencer works. I don't know all the deep, dirty details of that, but it's pretty amazing that you can get reads to 15 kb. Okay, so this is great for de novo assembly of genomes that you don't know about. It's great for RNA splice variants that you don't know exactly where the splicing is occurring. The error rate is 11% per base. Okay, so that's high. So you're not going to call a lot of mutations there, but the Illumina sequencer is like I think it's 0.01%, so that gives you a large difference. So the way that they get away with this error rate is they actually just sequence this same strand over and over again, and that's how they get away with this. And it's just like interrogate over and over again the same strand, and you can, even though the per base error rate is high, you can get sensible calls on one strand of something. But you only get 50,000 reads per run. They have made a bigger machine that I believe Mount Sinai will be running that does, I think it's in the close to a million reads. But this is a very powerful platform for different applications. I think in the RNA space it's pretty large, and de novo assembly of new genomes. So a lot of people are trying to use this for whole genome data with Illumina. So they'll do, like, the repetitive elements are very hard for the Illumina sequencers to see. And so they'll try to map the centromere with this, and then map the rest of the genome with Illumina.
So do they have more colors to distinguish? Like, how is it that they can map repetitive sequences?
So, so the main issue with the repetitive elements on the Illumina, which I didn't talk about, was when you make clusters, it's PCR based, and so repetitive elements do not PCR very well. So what happens is those clusters just don't form, we think. So PacBio, so only the single molecule sequencers don't use any kind of amplification for sequencing. So only PacBio and only Helicose can see the repeats with greater fidelity. So That's the only reason why we found the repeats in the cancers, because of the
And what's the
helices.
difference in error rate between next-seq and high-seq? You said
I
there's
think it's like,
higher.
I forget exactly what the exact percentage is, but I don't think it's that high.
It's not that much.
It's
So it's
not,
not
it's really not.
I mean, maybe twofold higher, maybe, but less than that.
I don't know. I haven't really looked into it in deep detail because I'm not interested in mutation or SNP calls. But a lot of the mutation SNP call people are like, we're still high-seq centric.
David, have you heard of Oxford Nanopore?
Yeah. That's the next one after this. So Ion Torrent's the other one. This one is actually CLIA certified. So Ion Torrent's in the clinical. They have probably the largest number of clinical ready tests in the clinic. So this one works by when you add an A or a T, it detects the protons being emitted by your nucleotide that you're adding in. And so it's not imaging, it's kind of a digital readout, but it's basically sensing pH, right? So it's super fast, okay? Super fast, like super fast, but crazy high error rates. And so that's what's hurt them from the mutation standpoint, but they sort of, I think their argument is you can just sequence more. Much faster, so you just have higher resolution by just sequencing more. So that's Ion Torrent. John Iafredi, I think, has one of these. Like I said, there are some clinical people, and Thermo Fisher owns this now because they bought ABI and Ion Torrent. All right, so Oxford Nanopore. People have been talking about this for like 5 years, maybe. So the fact that no one's using it that I know of is
No, they've changed a lot of things in the last few months.
okay. Well,
So Toshi has a system already. He got a system.
oh, he got a system.
Yeah, no, they've completely apparently changed their, like they have managed to figure out the diameter
They
issue.
figured it out. Okay.
Yeah.
Alright, so anyways, they detect current changes. So this is a concept that I remember hearing about when I was in college. So people were trying to sequence the genome with this type of technology. So the physicists were at Harvard and at MIT. So this concept's not new, but they would just detect capacitance differences of the different nucleotides as you go through this pore. So each of the bases have slightly different capacitance, and so it's a massively digital readout, and they were like, the physicists were like, oh, we can sequence the genome in like 2 seconds. So, I was undergrad a long time ago. So, they've been saying this for a while, but maybe they've changed it.
Yeah, no, they have. I think it looks like,
Maybe.
and what's really awesome is that they're, the sequencer.
You mean the laptop thing? Like
Yeah.
you just plug it into your laptop?
It's less than
like-
$1,000.
Yeah, so the sequencer would be really cheap to manufacture because you just need this core thing to work, and apparently you can just plug it into your laptop like a USB drive. And so you can just like fly to Africa and go sequence some stuff and just fly around with it. So it seemed really cool, but people like have really been, I don't know. So I've contacted them a couple times just to see if they would want to like play around with us using it, but it sounds like Toshi's actually gone.
Yeah, I know, they also have a deal, because Toshi told me that right now if you buy, they give you the machine and, how to make your libraries and everything. The whole package is less than $1,000 you get from them.
Right.
And you can run it and see how well it works.
So maybe that's cool. This would be agnostic to repeats, so maybe I should look at it.
Single molecule, right?
It's single molecule, very fast. Could be cool.
No amplification required.
And
Amplification.
so all you would need then is your nucleotide path if you're looking at it.
Yeah. You just
Nothing
fly
else.
to, probably just do it outside while you're like at the sandwich shop.
Might as
How does
well,
it
yeah.
go across the pore? It says there's some sort of potential across the membrane
I
or?
don't know. But right, it's like, it seems so cool, but at the same time, it's yeah, exactly. Yeah, but it seems really cool, and if it's actually working, which, if Toshi's got it working and he's sequencing.
Well, they were
everyone's genome
testing
right
it
now.
as of the last, about a week and a half ago when we met. One of his techs was going to set this up and said that if you guys want to play around with it, you can. And they were going to actually start sequencing using Illumina versus the Oxford Nanopore to see how they compare and how they're different. So I think.
Oh, they're going to be very different, but.
Yeah, so we'll see. I mean, they have it in their thing, so we'll see.
Yeah.
Yeah.
I think like some so the applications I think would be great for this would be single cells. Like if you can take a single cell and just sequence it natively, and there's no amplification. See, the other things are the repeats, but you know I'm slightly biased on that. And then there's chip-seq low input. You know, you just chip it and throw it on this and see what you get. So.
And you can sequence very long pieces too.
Yeah, it's just like PacBio, super long. Oh yeah, I heard the error rates. Oh, I don't have the error rates. So Illumina's high seq is like 0.1. I think the next seq is like 0.3 or something per base. The ABI was tenfold better, but that didn't matter because it died. And then the sample inputs were variable. I mean, the helicose had this advantage of putting in like basically sub-nanogram. And we didn't quite get to a single cell, but it was, pretty good for that. It just tells you, like, there are all these different technologies that keep popping out, and some of them have died. But you do, Oxford Nanopore is one of those ones that, is catching some fire, but could be useful for cool experiments. How to analyze that data, I mean, those are all the difficult things. Once you make a sequencer, you need to have the aligner and all these other, you guys have learned it for the Illumina because like literally, thousands of people have used it. So yeah, so mutation detection, ABI is dead, so it doesn't matter. So it's really Illumina. Splice variants, translocation. It's really Illumina long read pair end. Maybe you can make a library that's 500 base pairs and do pair end 150 on both sides and try to see if you get it over the translocation. But PacBio is probably going to be much faster at finding that. Novel RNA, it's total RNA seq is sort of what people are doing now. So this is like ribosomal depletion followed by random primers. With strandedness, PacBio is obviously another. Repeats my favorite. Helicose and PacBio, but Helicose died. And then limited materials, really, it's Oxford Nanopore is probably one of the most interesting ones right now. So I'm just going to go over some RNA-seq stuff. This I already showed you. So this is how we did single cell amplification with the ABI. It's just conceptually, it's fairly straightforward. You have polyadenylated mRNA, and you use an oligo-DT sequence, and we just created a universal primer sequence that had been used. And then you do first strand cDNA synthesis, and then you add poly A tail onto this with terminal transferase with DATP. And then you come in with another oligo-DT on this side. Now this side, the RNA has been degraded at this point. This side, all the A's are T's, right? So your oligo-DT will only bind to this side. Universal primer 2 sequence, go across the other way to fill in for double-stranded cDNA. And then here you shear. So we used to do ultrasonic shearing with the cobars, and this provides you the right size of amplicon. And then the first thing you amplify using these two sequences. So that was the idea. You have universal primer sequences on both sides. You would be able to amplify evenly across the transcriptome because everyone has a UP1, UP2. You shear, and then you add your adapters and then sequence. So that was sort of the more straightforward way of doing it. And then Illumina with CloneText developed this, where it's this template switching enzyme. So basically, it's still oligo-DT. It goes first strand. You add this smarter 2 nucleotide on this side, and the RT actually switches templates and goes the other way, from my understanding. And it's the same thing. You got this sort of universal primer sequences on the ends that you can amplify with, and that's how you amplify. But this template switching thing is their main thing that they have an advantage over the ABI single cell probe. So this was like a homebrew kind of thing. So I think we ordered 77 components to this to make this work. Well, this is now just like a kit you just buy, so it's a lot easier. And it works. It actually works, I think, better than the ABI thing. The one difference is, which I don't have here. So here we sheared and then cleaned up the ends and then added our adapter ends. So it's basically inserts itself around. So it's not. But some people make arguments like, well, if you shear mechanically, it's a little more random. Transposases may have some selectivity for certain sequences. I don't know if that's true, but that's. But everyone does what we call the Nextera prep. Nextera XT is for low input stuff, and Nextera Full is sort of for standard prep. So I think most of the Illumina people, or most of the sequencing people, are really using Nextera now, and not really Kovar shearing. And then this is total true-seq stranded RNA. So it's you start with 100 nanograms to 1 microgram total RNA. You deplete the ribosomal RNA. It's basically a bead-based thing that sort of pulls out all the ribosomal RNA sequences. Here you fragment the RNA here. So you fragment first. Remember the other ones that would fragment the cDNA. Then you go to first strand cDNA synthesis, second strand. Then you do this thing called the spry cleanup, which I'll explain in a little bit. And then you, there are all these other things where you just adapt the barcodes. Not so important. The main difference I wanted to highlight here is that the other sort of preps you do cDNA and then fragment. This one you fragment the RNA directly. So some of the RNA people would be like, well, that may be a little different than making cDNA first. These are all things that one needs to know as you're either submitting it or doing it yourself. Like, these things can affect how your sequencing output is.
But the great thing about this is that you're not dependent on polyagent.
Right, exactly. So that's I've become more of a fan of this because I'm into non-coding. It's stranded, so the adapters are put on in a certain orientation, so you know which strand the cDNA came from or the RNA came from. And so it can tell you, like, are you in a given region, you can actually know, are you in the reading frame that's open, or are you in the antisense direction? And the antisense is how hot air was found, right? So hot air is an antisense and a coding gene. So these are all the big things to consider. Oligo DT based stuff is like 3'. It's usually 1 cDNA per transcript. So when you normalize this kind of data, I think my argument would be you would do reads per million because it's one cDNA per transcript. Because it's oligo DT based, because there's only one poly-A tail. When you do random primers, you got more coverage of the transcript, but you need to think about normalization of length. So this is where they use RPKM. I don't know if you guys have heard all these terms already. And something that people don't think about is that you may align to the genome, and then when you normalize, like, you may see funny things in some of your samples because your ribosomal RNA depletion may not be complete in all samples. And so if you don't take that into account and subtract the ribosomal RNA computationally, you can totally affect your normalization, because all of a sudden, just because of your technical issues up front, your normalization is all screwed up because you have a bunch of ribosomal reads in some samples but not others. Does that make sense?
Does the spray cleanup help with that?
No, not necessarily, because this would occur because you're making cDNA. So these are like little things, but people don't necessarily think about it. You just get your data, you align, you're like, oh, I normalized to all alignments, and oh, wait, this sample had 20% ribosomal contamination, and this one had 50, and that's just because, it's not 100% perfect. I think everyone can see that.
So how do you deal with that situation?
You have to just basically remove those ribosomal RNA counts digitally, right?
Yeah.
And then you normalize to the rest.
Okay.
Right, because it's sort of like a digital ribosomal depletion.
Yeah.
Which you would have hoped that your ribosomal depletion had done it 100% efficient the first time, but you know how it is in each tube.
Yeah, totally. This is a very important point for us.
Yeah, little but big. And then here you can see these non-polyadenylate, non-coding RNAs. And so that can affect how you normalize things, because now all of a sudden you have all these other reads from other places, and how do you normalize? It's not just polyadenyl mRNA anymore. What's the right type of normalization? This is where people start getting into these, you should spike in synthetic RNA and really have sort of quantitative transcripts. So you can like spike in a certain ladder of RNA and look for that ladder of RNA in sequencing, and then you can sort of translate samples to that ladder and then have a true quantitative metric to compare each sample. The other sort of way to do it on the more extreme side is to do molecular barcoding. So people will put barcodes on each cDNA transcript or on the library standpoint, and then you then have, like, you could put 100 barcodes on one side and 100 barcodes on the other, and now you have a complexity of 10,000 universal molecular indexes. And so then you say, well, there's more than 10,000 transcripts in the cell, so the way they deal with that is that, you know, it's unlikely that you'll have 10,000 transcripts of one type of RNA. So you take the universal barcodes, 10,000 combinations, plus the unique sequence of the transcript itself, and it's unlikely that you're going to have 10,000 gap DHs. Maybe you will, but you know what I mean. So that's how universal barcoding works. Molecular indexes work. So that sounds really cool. The computational people love it. They're like, oh, you should like UMI everything, because for them it's perfect. It's like, oh, it's like one unique transcript that you've made a library on. But the problem is that UMI barcoding process is not 100% efficient. So we know that you're probably losing data from the barcoding itself. So I tend not to barcode, but these are just things that all of us have to, you know, make for our own. You have to decide on your own. Amplification. This is the amplification bias stuff. Yeah, we see some of this, but it's not, to me, from my standpoint, I haven't seen at the single cell stuff. We see amplification seems to be somewhat predictable. There is some bias, but I don't think it's as drastic as people make it seem. And then this stochastic loss of low abundance transcripts. So when you're at like really low abundance Poisson statistic kind of sort of transcripts, amplification is going to be highly variable. So people are like, oh, I really want to focus on this really rare transcript in my single cells. Yeah, that's kind of tough. But you know, if you do a lot of single cells, you can overcome that. Fragmentation size. So some people don't think about the amplicon size that you're making. So if you make, I think the total RNA-seq is like 200 base pairs. If it's 200 base pairs, then 150 pair end is kind of useless, right? Because you're getting 300 reads of, you know, a lot of them are overlapping. Now, if your amplicons are bigger, then you can look for translocations a little easier. So those are all things that are customizable that people, if you just do it on your own, can customize. But most of these kits, you know, it's just one way. But you know, that size of amplicon can affect whether or not you see a certain translocation or not, or if you see, if you're looking for really short stuff, you know, the size of your amplicon is important. Total RNA-seq, mRNA-seq, I think you guys kind of understand that, so I'm not going to go. Stranded RNA-seq, we just talked about. And then single end versus parents. This is really like, you know, I've been sort of talking about this splice variant translocation thing. But basically for Illumina to see it on a pair end, read one may be over here, read two maybe over here. I mean, the other pair on this side. This one's your perfect one. It's got one on both sides. So this one's usually not a problem. This one you only see one half of it. This one you only see one half of it. But you can see like the chances of getting this 100 times is pretty hard for this transcript. If you hit this, you're like, oh, you should be able to see this. But the computational people, at least the ones that I haven't seen them figure out how to deal with this. If you have a read that's really split in the middle, because what you would have to do is try to align like 15 base pairs here, 15 base pairs here, 15 base pairs here, and try to look for these. And you can imagine if you did that for every read in your pipeline, how slow that would be computationally. I'm sure there's more computationally sophisticated ways of doing that, but I haven't seen it.
But you can look at all the unmapped guys, right? And just so that would
Right.
be
So you can look at things that are not uniquely mapped, and then filter through that. But then you have to like tell the aligner, okay, I'm just going to take the first 15 base pairs of this one 50 base pair read and align, and do that to the other side of it. You know, I don't think there's a straightforward. There hasn't been a pipeline, and that number can be variable, right? So you know, computational play. Well, you know, you can do like 14 and 17 and keep changing it around, but then you have these, you know, sort of alignment issues. So, so just a little bit on RNA DNA purification. This does matter because it affects like your library construction. So SPRI is, it's basically, it takes advantage of charge, the phosphate charge on DNA and RNA. So the longer your DNA, the more phosphate, the more negative charge. And SPRI, I don't know all the details of the chemistry because some of it's proprietary, but it's basically sucking up positive, it uses positive charge to suck up DNA. So the more SPRI beads you put in, it affects the ratio of some kind of ethanol isopropanol, similar to what you would do for trizol, you know, precipitation of RNA, and that crashes the RNA or DNA onto the bead, which is keeps it bound. So the more SPRI beads you add, the sort of lower molecular weight stuff you can pick up. So some people are like, well, you know, for my libraries, I don't really want to try to, I don't want this junk up here, and I don't want primer dimers down here, so we can do this thing called double SPRI, which I don't think we need to do anymore, but we used to do in single cell preps, where you can actually add like 0.5x SPRI, so, and like take the supernatant. So the supernatant would be anything below basically like here 500 base pairs. Okay, so you would take the soup, throw out the beads, and then you would take that soup and then add 1.8x SPRI, and so you would pick up everything that's small, but not too small. So that would clean up the primer dimer piece. So that's what we would call the double SPRI. So this is just a way to purify without, you know, the old way we used to do is run out of gel, you know, cut it out. Maybe none of this. Maybe some of you guys remember this. Right, you cut it out. You're like, oh, I'm gonna put this little piece of my gel on this column and melt it, and it's gonna work. And sometimes it works, sometimes it didn't. So that was the old method that some people were still trying to do. And this is, I think Toshi's got this machine with wet steam. So
I've also
huh?
chipped in that one too.
Right, yeah, so this is, I think it's kind of a cool thing. We didn't need it because we were using so much SPRI. But basically, it's gel electrophoresis, and you run like a ladder with it. And then you tell the machine, I want this fraction. And it basically squeezes, it basically uses this charge down here. It sort of allows the DNA that's in the gel to go into a collection chamber, just based on like location. And so there's no ethidium bromide, so there's no intercalation issues. And it's basically cutting out your gel, but cutting it very well. But it doesn't cut, it just sort of sucks it out.
The only problem with this machine is that you can only run 6 samples at a time.
Right.
First thing. Second thing is that it takes a long time to run it. So, and also you have to buy a separate cartridge for each 6 lanes that you run. So overall it's cumbersome. We, still in the lab, actually cut out bands and process stuff that way. If we want to do that, not for all of our samples, but when we were sequencing microRNA size RNAs for our
yeah, so sometimes when you're dealing with microRNA, you just gotta cut it out, because SPRI doesn't work for tiny stuff, and this would be impossible, I think, to narrow down your window. But anyways, so the point is that this machine is around. It's in the Kansas Center if you want to use it. So most people I think that you want to use this are the chip-seq people. So you have like low input chip, you don't want to lose any of it. You try to separate it on this. SPRI may not be able to retain, not be able to collect them at the sort of size that you want exactly for sequencing. So I think that's why wet stain is obviously went for this one. But for us, like since we do so many samples, like the throughput was like impossible. So and so can you make your own library? So this is the P5, P7 sequence. So P5 is this one. I think P7 is this one. And so those bind to the flow cell. There's some spacer sequence here. This blue sequence is the read sequence. So this is the sequencer adds this primer to initiate sequencing, you know, just like you would do in the capillary sequencer. And so the sequencing will start right after this. Okay. And so, you know, you can actually just synthesize this and add your adapters for either barcoding or sequence-specific sequence, and make your own Illumina custom library. I guess you've tried this. Did I send this to you?
No, you didn't send this to me. I got that information too, but my point was that that region that's not underlined,
This one here.
yeah, that is actually really important.
Woo!
Yeah.
Anyways, so I've done this before for custom applications. But the point is that you can just do this, right? You synthesis from IET, say you want to just like look for translocation, intrahepatic cholangiocarcinoma or something like that, right? And then you can basically generate probes against one side or the other, PCR, or RT-PCR, and then try to sequence it to sequence confirm, something like that. So one of the things we were doing was like line 1 seq, which is basically looking for that retrotransposon. So on one side you would look for the line 1 sequence, and the other side was random hexamer. And so you would try to map all line 1 insertions in the genome. So people have done that for cancer and germline, and we tried to do that, and it worked okay. But that's the point. So if you guys ever want to go make your own stuff, if you want to look for certain things in the genome or certain RNAs, it is possible. Alright, and then this is my last couple things. So you guys went over red squid. Okay, so I'm not going to talk about this, but the point is here is like all of our sequencing looks like this. Like the beginning of the bases look great, and then some of them go down here. These guys, this one will look great all the way through. And so this is very important, because if you call a SNP or a mutation out here, there's a chance it might be wrong. And very good scientist, Howard Hughes, who's in science. And they found all these RNA DNA differences. So what they did is they had RNA-seq and DNA-seq, and said, wow, there's all these differences, all these variants. And that's what was published. It was pretty awesome. They called them RDDs, and then they verified that some of these may be protein coding changes. So then there was 3 subsequent rebuttal publications in Science, or comments, that all came out at the same time, the same issue, and basically saying this is a sequencing artifact. So, oops. So here's position and alignment, so 0, and this is out to base 50, and then the mismatch was occurring, a lot of them were occurring out here, or on this side, which is probably on the opposite strand, or the pair end. So the points where they were finding all their hits at the ends of sequencing. And so those are just sequencing artifacts, unfortunately. But, you know, she's still, you know, they're still scientists, and it's just a mistake, but it's a little scary, right? Because you're like, you just trust the ATCGs that are coming out, but if you don't think about that, the end of your sequencing, there is higher error prone. If you're trying to look for SNPs, this is the risk that you have to take into account. And the computational people, maybe some of them are taking into account for you, or maybe not. But if I think it's important for you guys to know is like if you start seeing something that looks like a massive number of variants that are occurring, like maybe it's a sequencing error thing. That's not necessarily something you guys are going to make a pipeline for, but something to talk to your computational friend about. And then normalization. So this is single cell data, and I just wanted to show this just to show you that like amplified stuff. This is 5,000 picograms in this paper. This is 500, and this is the same sample technical replicates. And as you get lower and lower, sort of the correlation of your data. This is where the amplification bias issues people are starting to talk about. And this is our data, bulk RNA, when we were having these kinds of funny issues, okay? And this is just reality, because this is 10 picograms of RNA. And this is single cell data. You know, it was impossible to do this, because where they're, this is individual single cells. So at some point we try to take one cell and split the RNA in half, and then amplify it. It'll look as a total mess. I'm sure. You probably did that too with the bead, right? So, and so this is just to show you, like, you know, different types of samples, even when you do single cells or bulk tumor, have different what we call dropout rates, so the percentage of genes that are absolute zero reads. And so you can imagine how this can impact if you do differential expression, right? You got a bunch of zeros in this type of sample and not so many on these. You're going to have a lot of differential expression just because of zeros, not because it's necessarily true biology. Like you may have like 10 transcripts here and 0 over here, right? So, and so when we did unsupervised clustering with just reads per million, what we were seeing is this kind of phenomenon. We call it the bursty expression phenomenon. So this is like just reads per million normalized, and so you would see like certain genes being dominated by the single cell transcriptional profile for each of these single cells. And so even though they clustered, it was like this is looks a little funny. Something funny is going on here. We ended up doing something that's basic in microarray data, which is you take the median across all samples, and you what you do is what they call median polishing. And so you sort of try to normalize the data for every gene and every cell, and the heat map looks better. So my point is that the way you normalize can make your data look like this, or it can make it look like this, and that's it. This is just normalization differences, and that led to all these things that we were able to do. But and when you do differential, you guys learned DE-seq too from him, right? So
Monday.
Oh, Monday. Okay. So this is if you took that single cell data and do DE-seq, just like tell DE-seq to find stuff. This is what DE-seq found, and they were saying that all these genes were differentially expressed, but there's no clear thing here, and this is because of single cell data, because of all these zeros, these are these things, and so even though DE-seq we're saying these are significantly different between CTCs and primaries, I don't know about you, I don't see any difference. But this program said there was. So these are the limitations of computers and not your eyeballs to see differences. And these are all these like individual genes we looked at, but basically there's like no difference. And EdgeR is another differential expression package on the same type of data. It's like even worse. It's like there's like no difference, but these EdgeR was calling these as significantly different. Okay, so this is just a volcano plot t-test, and this one does a slightly better job, but
So for single cell data, what would you recommend?
so that's what I would not recommend.
Just a straight
No. So I think some people have tried to, with Ben, have started to do t-tests. The thing that we ended up, Ben really, what we realized was if you just do what we call non-parametric, which is ranked products, so in the statistical world, it's like Wilcoxon. So you just rank your gene based on expression within the same sample set. So like 10,000 transcripts in one cell is the top gene, but in another cell it's 9,000. You don't really care that it's 9,000 versus 10,000. That has no meaning, but their order of the genes matters. And so that's what you would do here. So the probability of having that gene at the top 3 times, and you basically multiply it. There's a reason why the product of these ranks is more important than the sort of sum. So the rank sum is the Wilcoxon thing, but I'm not a statistician. But conceptually, what it is basically you're just ordering genes relative to each other across samples, and that's what led to being able to. This is all the statistics stuff. So that's what led to this ability. So this is what rank product was telling us was different. So anything in purple was high in CTCs, and yellow was high in tumors. And you sort of see when you look at it visually on the heat map, they sort of match up where they should. So we knew that this statistical method was able to see differential expression, while the standard things that you would get at bioconductor Broad not able to do it on this type of data. So
Was this Illumina?
this was all ABI, but it's the same on the Illumina data. It's the same. You
I've
get
seen
these-
crazy stuff with EdgeR.
Yeah, EdgeR is like, what is it? I don't even understand how it comes up with this. I mean, look at this. I mean, this is like ridiculous, right? And it's like, how is this different? I don't see any difference here.
Barely.
Like EdgeR says it's upregulated. Really? You know, statistically it's upregulated. You know, so you know, I think that's the, you know, as you're like learning all these methods, you have to realize like, you know, a lot of what you need to do up front in terms of normalizing your data will significantly impact how those different expression programs work. So like this type of single cell data, you know, a lot of people want to do single cells. You can't use the standard packages, and I don't know. I became very partial to this ranked product thing, probably because it worked, but you know, other people are trying to just use simple t-tests, and the simple t-test is like you make these volcano plots. You just look for things here versus here. It's like it's like the student t-test, right? It's very straight up. So I often do that on my own. So sometimes I just take all the reads. If I don't want to do my bioconductor and mess around with code, I just put it in Excel. I will just calculate the t-test. Excel is able to calculate t-test between two groups, and you know, sort of just do a litmus test. As you know, does this data make sense to me? And that's partially because of my, you know, when I was a chemical engineer, and at that time at MIT they were phasing out what was it, Cobalt, sort of old programming language, and so they were actually training all of us on Excel and how to maximize statistics on Excel, just because at that time that's was happening. So I think R was in its infancy and whatnot. But anyways, that's all I want to try to say to you guys is you have to take into account all the sequencing library construction for anything you're doing. You have to take into account how you normalize your data, and you have to take into account like these programs are not as smart as you. Like your eyeballs are probably better than the program. So I always tell people always look at the raw data, because if the raw data doesn't look like what you're seeing by your computational method, there's probably something wrong with your computational method. Does that make sense? We see this a lot with normalization. So sometimes like DEC2 will take transcripts that are very low and then artificially inflate them, and you wouldn't have known that unless you looked at the raw data. So sometimes just doing those litmus test things, you know, you'll avoid having some issue down the road. That's all I have to say. Any questions about stuff? Is that informative, non-informative? I don't know.
What would you recommend doing median polishing normalization?
I think when you have like the sort of like expression patterns are like don't look like they're consistent across samples, you can median polish, and that helps with just normalizing your data. That's what they used to do with microarray data, because each microarray would have like slight differences, and so they would basically try to normalize to the median for every microarray. And so.
And then you take that and you do your rank ordering based on that?
Mm-hmm.
Your median, right?
Yeah. Median polishing and the purpose of this was, you know, was really for making the clustering looking. Heat map look cleaner, to be honest, because the samples did cluster, but it was like it was a bit of a mess. So, and that's what happens when you get like lots of samples with different sequencing depths, and you don't know that that's true biology or your sequencing depth. And you know, I think that's why like a lot of times they say like you know, I hear like some people want like 30 million reads per RNA-seq. It's because they feel like that's a lot and you should have enough, but can you get away with 100,000 reads? Probably. I mean, you know, I think Aviv single cell data, I mean, you were working with Aviv on this, right? You guys were doing how many, do you remember how many reads you were down to?
I do not remember.
Yeah. But it was like
per
10...
Wait, per cell?
Per cell, right?
I think it was like...
It was pretty low. It was
yeah.
definitely less than 1 million. But they were able to extract enough information from like, but they overcame it with just doing massive number of cells. So, but they would like take 100,000 transcripts or reads per single cell, but do like 10,000 single cells, and then they could see the patterns just by having, because you don't have depth of coverage. It's estimated that we have 500,000 to 1 million transcripts per single cell in each individual cell, but even that varies. So like a lymphocyte versus a tumor cell, the number of transcripts is probably different. So the single cell thing, there's like all these normalization issues that people bring up. Like I think Rick Young at MIT really thinks that there should be controls put into every single cell, so you can quantitate absolute copy number of transcripts in each cell as you compare them. Because the assumption is that, you know, when you sequence something, you try to normalize it based on your sequencing prep, but the reality is that that's maybe not the same in the cell. Like a lymphocyte may have like 100,000 transcripts and a tumor cell has 1 million. So comparing them when you put them up to the same million is probably not accurate, but that's the Rick Young cell paper that they put out. So all these like philosophical, mathematical questions, but especially for single cells. But even for RNA-seq, it's the same thing. You know, like lymph tissue versus tumor tissue, the absolute number of transcripts is probably different. I mean, it makes sense, right? But the assumption when you do this type of analysis is they're not different. Because you just normalize by total reads, which is the sequencer, not the biology. Does that make sense? I know it's a little strange, you know, the normalization thing, but the normalization thing is a huge thing. But it, you know, so if you're asking some of these questions, you may lose a lot of your data because the truth is really there, but it's you lost it during normalization.
You just normalize within, you know, just something within the cell.
Right, so that's what Rick Young's argument was for cell paper. You would put artificial RNA in each single cell.
Oh, but not even something artificial, but you couldn't do something that was native already in the cell.
Right, because you don't know we don't know what the truth is.
You
So
don't know what the answer
yeah.
is.
So the truth, so he would say like the truth is when you put in an artificial ladder and then amplify at the same time as your single cell, and then you can sort of equate. Now, we started doing that, but like we would occasionally see that like the ladder didn't get in, or the amount you pipette is maybe a little off, you know, because you assume that you're pipetting perfectly in every single cell. But you know, I think it's not unreasonable we started doing that, and we sort of realized that at the end of the day, like it may be important for seeing stuff at the very low end of your transcriptome, but for the high expressors in the medium, it didn't really matter to sort of see what was different. That makes sense.
All right, let's thank Dr. Ting.
Thank you.
Thank you.